Today I will be talking about synaptic or neural communication, which is the communication between neurons in the nervous system and between neurons and effector tissue throughout the body. So the first part, the functional unit of the nervous system, is the neuron or nerve cell, and it has many different parts. The large part that you see here, which contains the nucleus, is the soma or cell body, and the branch-like protrusions off the body or soma are the dendrites, which are receptive ends that will take in signals. Usually the longest part of the cell is the axon, which begins here at the axon hillock, or the part of the cell body which protrudes in a cone-like fashion to make up the neuron, the axon. And the most axons are myelinated, or they contain myelin sheaths, which help to insulate and protect the axon and the rest of the nerve from electrical energy, and they also reduce synaptic delay, or the time that it takes for an action potential to travel all the way down the axon to the other, to its target. So, and at the bottom here, we have axon terminals, or another name for those are synaptic knobs. And we classify, and one of the ways that neurons are classified are by their function. They're sensory neurons, or afferent neurons, that take in information from other parts of the body and send it directly to the nervous system, or the central nervous system. Motor neurons, or efferent neurons, send messages to muscle cells throughout the body. And interneurons, or associated neurons, are neurons in between neurons, so ones that will be relaying the message between other neurons. And the way that these neurons send messages is through action potentials, which are short electrical impulses that occur by switching the polarization or the charge of the plasma membrane on the neuron. This also occurs in muscle tissue, but we talk about it most commonly as referring to neurons. And that occurs because normally charged particles like sodium and potassium ions will not be able to cross the phospholipid bilayer, but they can when transmembrane proteins known as ion channels open up, and they can diffuse normally across their concentration gradients. In order for this to happen, certain qualifications have to be met. The resting membrane potential is what we call the membrane potential at its normal state, and that's about -70 millivolts. And as this changes, or as some of these channels open up and sodium and potassium ions are allowed to flow inside, the membrane becomes depolarized, or it switches polarization, so that generally the positive end in the extracellular space flips with the negative side, and this causes electrical signal to be propagated down the length of the plasma membrane. And it does this by something known as the all-or-nothing principle. And basically, the resting membrane potential, or the transmembrane potential, has to reach -50 millivolts all the way up from -70. Otherwise, an action potential will not take place. And in synaptic communication, there are two basic types of ion channels. One of those are ligand-gated ion channels, or chemically gated ion channels, which will open up to allow ions pass through them upon binding to some sort of chemical receptor, or chemical signal or ligand, like a neurotransmitter or a hormone or other ions. And so, in this case, when a chemical binds to the portion on the receptor portion of the transmembrane protein, it will open up or do a conformational change and allow charged particles to flow through them without being blocked by the phospholipid bilayer. The other type of ion channels are voltage-gated ion channels, which will respond to changes in the transmembrane potential of neurons. So as it becomes more positive or negative, these might open up or close to allow or stop the flow of ions to the intracellular and extracellular spaces. One type of chemical, or that we're specifically talking about when we're talking about chemically gated ion channels, are neurotransmitters. And neurotransmitters will be secreted from the axon terminals and go to receptors on either whatever type of cell that they're going to or another neuron. And we classify these broadly into two categories: excitatory neurons like acetylcholine, which cause something to happen on the end that they're received on, and inhibitory neurons like glycine, which will act to stop something from occurring. And so a synapse is the part of the area that we're talking about that in which communication is taking place. And there are two basic types of synapses. One is electrical synapse, which basically just has gap junctions through which ions can travel to other neurons. And the other type is a chemical synapse, like the one shown here, wherein as the action potential flows down the axon, neurotransmitters contained in synaptic vesicles can be exocytosed, or the vesicles will bind with the plasma membrane on the axon terminal and be excreted out into the synaptic cleft. And as you can see here, the dots represent neurotransmitters, and they can be picked up by dendrites of other neurons, or they can directly be picked up by even the cell body of other neurons. And to clarify which neurons we're talking about, we use the terms presynaptic and postsynaptic. In this case, the presynaptic neuron is the one sending the chemical signal, and the postsynaptic neuron is the one receiving it. And so that's why this would be called the postsynaptic membrane, and this is the presynaptic axon terminal. And the synaptic cleft is the space in between the synaptic knob and the cell that's receiving the information. A specific type of chemical synapse is one revolving the neuromuscular junction, or where muscle tissue is innervated by neurons so that they can contract. And in this case, the synaptic cleft is in between the presynaptic the presynaptic knob and the sarcolemma, or the cell membrane of muscle cells. And so in this particular case, as the action potential flows down here, it will open up voltage-gated ion channels and allow calcium ions to flow within the cell, and that will cause synaptic vesicles containing acetylcholine to bind with the plasma membrane of the neuron and exocytose the acetylcholine into the synaptic cleft, which then diffuses across the synaptic cleft onto nicotinic cholinergic receptors in the muscle tissue, or the cell membrane of the muscle cells, and bind to ligand-gated ion channels on the membrane of the muscle cell. And when this occurs, it will cause ions to flow within the muscle cell, and that will cause voltage-gated ion channels across the plasma membrane of the muscle cell to open up and allow an action potential to go across muscle cells, which is another type of tissue that is excitable and can perform the propagation of an action potential. And so when that goes, or when the action potential occurs along the muscle cell, it will go down the transverse tubule and to the terminal cisternae, in which case more voltage-gated ion channels will open up and allow calcium to move on so they can go to the sarcomere and cause muscle contraction. Oh man, I'm feeling so